seconds.
Welcome everyone to Iris Biotech's 5th online workshop. True to our slogan, Empowering Peptide Innovation, we are keen on knowledge exchange and getting in contact with scientists all over the world. Therefore, we started this workshop series last summer. Today, our focus is on polymer therapeutics for improved drug delivery, surrounded by some brief introductory information about the company Iris Biotech. My name is Raimund Meyer. I am working as a project manager at Iris Biotech, and today I will guide you through this online workshop. As you see, I am not Karin Rustler as announced as a chair for today, but unfortunately our colleague Markus is not available today, so we had to shift a bit the roles. If you wish to announce your participation on social media, please feel free to use the hashtag Iris Workshop Series, which is also displayed right now in the Microsoft Teams chat. To have a pleasant experience for all participations, I would like to begin with some technical guidelines. To ensure the best possible audio and video quality, all participants are automatically muted and your video camera is switched off. Another information is that this live event is recorded. Thus, if you enter late or miss anything, please check on the bottom left side of your Teams screen if you are watching the recording or if you are live, which is shown by the red broadcasting live symbol. You have the possibility to pause the video, rewind or jump back to the live session. In addition, the recording of the workshop will be uploaded on our homepage afterwards. Questions can be submitted by using the Q&A chat of Microsoft Teams. You can enter your name or submit your questions anonymously. We will frequently check the questions, publish them and submit them to the respective speaker. If you have any technical queries, you can also submit them in this Q&A chat. Our technical assistant will then connect with you and try to solve the problem. Initially, the questions are just visible for the organizers and will then be published to the audience. So don't worry if your submitted question does not appear on the chat screen immediately. To try out the Q&A chat, we will now post a question which you can answer in the chat. Now you should see the question, what is the topic of today's workshop within the chat? Please feel free to answer it to try out this chat tool. Yes, and we already see the answers coming in. Of course, the correct answer is polymer therapeutics. So as this seems to work, I would like to quickly give you an overview of today's agenda. We will start with a short company and topic introduction by our project manager Karin Rustler, and afterwards our external speaker Maria Jesus Vicente will provide detailed information on her research at the Polymer Therapeutics laboratory in Spain. As mentioned, questions will be collected throughout all talks in the Q&A chat and are forwarded to the speakers. Of course, you can also get in contact via email or phone call after the workshop. One last note from my side. If you accidentally close your Microsoft Teams window, don't panic. As this workshop is also available as recording, you will not miss anything. Just re-log in and view the recording. You can then also play this recording at increased speed until you catch up with the live event again. After this technical intro, let's continue with the first agenda point, a short introduction to our company by our project manager Karin Rustler. Karin studied biochemistry at the University of Regensburg and obtained her doctoral degree in organic chemistry for the synthesis and characterization of photoswitchable compounds for biological applications. During her PhD, she stayed abroad at Kyoto University, Japan for several months. Beginning of 2020, she joined Iris Biotech as a project manager responsible, among others, for the field of drug delivery and polymer therapeutic projects. Karin, thank you for your precious time. Please go ahead with your presentation.
sorry, I was muted. So now, thanks, Raimund, and also welcome to all the participants from my side. As announced, I will give a short introduction to the company Iris Biotech. Iris Biotech has been founded in 2001. Together with our partners, we are represented all over the world. In 2013, our own synthesis laboratory, the Iris Biotech Laboratories, have been founded. 1 year ago, in 2021, we refreshed our company logo and the corporate design. So in the middle of the page, you see the old logo, and on the more or less bottom left side, you can see the refreshed one. From beginning on, our guiding principle was and still is empowering peptide innovation. But what does that actually mean? We are providing a knowledge base with various brochures and flyers, as well as a bi-weekly newsletter, and we are attending conferences all over the world to get in touch with researchers. On this slide, you can see some of our refreshed brochures and flyers, which will soon be available free of charge on our website. Related to today's topic, I can highly recommend the brochure on Polymer Therapeutics, as well as our flyers on uniform packs and also on Polymer Therapeutics. Besides our knowledge base, empowering peptide innovation also means that we are supporting our customers in their ongoing projects as qualified supplier. We are providing our products from research scale to commercial production for universities as well as for industry. Besides, we are also pleased to act as consultant. So if you want to start a new project and want to discuss about suitable products, their applications, or if you have any other questions, feel free to get in contact. We are happy to discuss with you. Our portfolio focuses mainly on building blocks for the synthesis, modification, and purification of peptides, as well as on linker and drug delivery technologies. So this leads us already to today's topic, Polymer Therapeutics. But why do we actually need drug delivery technologies? So often a promising new drug is discovered, but then it turns out that the drug is either not sufficiently soluble, it cannot reach the desired target, it is attacked by the immune system, it is degraded by endogenous enzymes, or it suffers from rapid renal clearance. Drug delivery polymers can avoid the before-mentioned drawbacks by solubilizing the drug, preventing degradation, reducing immunogenicity, as they are shielding the drug, and also by preventing renal excretion by increasing the drug's hydrodynamic radius, leading to overall prolonged half-life. At Iris Biotech, we are offering various polymers as well as copolymers as listed on this slide. So this is just a brief summary, and with this, I would like to finish already my introduction and hand over to Maria to get to know more about her research. Thank you.
Yeah, thanks a lot, Karin, for giving this introduction. Now let's go on with our highlight and switch to the external guest speaker, Professor Maria Jesus Vicento Con, head of the Polymer Therapeutics Laboratory in Valencia, Spain. Maria received her PhD degree in chemistry in 2001 for her research on solid supports at the University Jaume I in Castellón, including 1.5 years research at UC Berkeley with Professor Fréchet. She then moved into more biomedically oriented research, starting with postdoc positions in Castellón and Madrid. And in 2002, she joined the group of Ruth Duncan in Cardiff, Wales. In the following years, Ruth and Maria really pioneered and revolutionized the field of polymer therapeutics. In 2004, Maria started her work at the Príncipe Felipe Research Center, Valencia, as a research associate. Was then promoted to her current position as the head of Polymer Therapeutics Laboratory at the research center, and also as a professor at the University of Valencia. Based on her research results, she founded very successfully the company Polypeptide Therapeutic Solutions 10 years ago. Until today, Maria's research group focuses on the development of novel nanopharmaceuticals for different therapeutic and diagnostic applications, in particular, the application of polymer therapeutics in unmet clinical needs. Maria Jesus has received several awards of excellence, and she organizes the biannual International Symposium on Polymer Therapeutics. Maria, it's a great pleasure to host you as our guest speaker. The stage is yours. We are looking forward to hearing about your research. And for the audience, please post your questions by using the Q&A chat again.
Thank you so much, Raymond. For me, it's really a pleasure to be able to give a talk to IS Biotech. You know that we know each other for many years, and since 2008, where I first organized the conference here in Valencia, you already were here. So you are very trustful people, very supportive people from the very beginning, and also helping us with the development and the growth of the PTS that I know you are still collaborating with them. So that's really, myself, it's really a pleasure to be here. And what I will try to do today, it will be to show some of the projects that we are running in the lab. We are a very active group based on Valencia, and we are really having highly interdisciplinary approach where we have people working on the polymer chemistry side or bioconjugation side, but also we have a lot of animal models and cell culture and so on. And we have all this interdisciplinarity because to really develop therapeutics of applications, we need to talk to each other. And because our major motivation is to reach the patient and to solve unmet clinical needs. We are mainly focusing on those unmet clinical needs because we need to find solutions for example, metastatic tumors or more and more CNS disorders that are more prevalent in the SHG population, because there are still some of these challenges that hasn't been moved in the last 20 years. And we have selected nanomedicines to really try to reach these goals, and in particular polymer therapeutics. And when we selected this path to reach our goals, one of the things that we realized is that we are working with complex structures. And with complex structure, you have to be very careful with the chemistry and with the characterization techniques. So the exhaustive physicochemical characterization is coming with us from the very beginning till the end. And only in that way, you will be able to control manufacture, scale up, and then to reach to the patient. That's why we really are investing our time from the very beginning in the design of a project to control the chemistry and the biology. Also, because not only is important to have well control the chemistry and the physics, but also which models are we using. So it's more and more we are implementing closer models to the patient, trying to have the answer of the question we are asking, because the preclinical models, all of them have limitations, but we are trying to really put in the goal in vitro and in vivo models that can reflect some answers to really move from preclinical to clinical status. But always we are really keen of having our conjugates in the patients, and always keeping in mind that the patient population is highly heterogeneous. And what we do in the whole path is trying to identify functional biomarkers that is allowing us to identify which patient population are going to be benefiting for our design nanomedicine. Not everybody will be able to be benefiting. Some of them will be more than others. So we try to understand which of the biomarkers or the biophysical predictors that really help us to better design in a personalized type of manner. And as you will see, all this, how it works in our lab. So we have this type of reiterative scheme, where on one side we have the importance of the identity of what we are synthesizing, not only by the compound itself, but also and solution conformation, how these nano-complex systems are present into biological membranes, because this bio-nano interface is the one that is going to rule all the possible interactions. So depending on how our solution conformation is acting, the cellular membrane interactions is where we are going to trigger a different therapeutic output, always keeping in mind that our patient population is highly heterogeneous, so we could see which one we are targeting the best. So we are implementing techniques like triple fractionation, for example, LCMS, trying to identify and to know the fate of our nanosystems in physiological fluids. We try to really not only identify the conjugates we are administering to the animal or the patient, but also the metabolites that are responsible for the activity. So we are monitoring both, and we are more and more focusing on these techniques that are validated by the PUNCL, or they are used afterwards in the manufacture, in process control. So we are trying to implement validated analytical techniques that are also able to be moving forward in the next steps in the chain. So we are able to have this AF4, LCMS, NMR, and even the process of synthesis and purification that we can really move forward afterwards. Not only with the chemistry, but also we are doing it with the models. We are fully characterizing all of them. We work with cells, 2D and 3D models more and more, even patient-derived models. We characterize for critical parameters regarding endocytosis, endogenous triggers that we can use to activate our prodrugs, and also how they communicate between them. And like we are also looking, for example, for exosome release, this extracellular vesicles communication. So we are characterizing our models in vitro and in vivo. For example, when we work in cancer models, we need to understand our tumors need to be orthotopic, it has to be metastatic, we need to know how well they are vascularized, how are the metastases progressing, which are the markers we can use, and if there is anything in the blood circulation or any physiological fluid that we can use as a biomarker. So we are really doing all these things at once, trying to move forward from the beginning to the end in a much faster and validated manner. And with all the strategy we are applying for different projects, we are working more on the basics type of things, or apply, let's say, on the materials area, where our materials can go directly to specific organs or organelles inside the cell. Some of them have inherent tropism to the kidneys or to the lymph nodes, for example. And we have projects that are targeting mitochondria, for example. It's a key organelle intracellularly. We are applying a lot of combination therapy because we are approaching highly complex pathologies like metastatic tumors or neurodegeneration. We are also trying to identify those biomarkers, and at the end we go to have breast and prostate cancer metastatic models. We have the last years applied also for neurodegeneration, as we do have platforms that are able to reach the brain bypassing the CNS, the blood-brain barrier, on pediatrics and also in Alzheimer's, but also in brain metastases derived from breast, for example. Or if we don't want to go systemic and we want to target something specifically topical, we have been working a little bit with the skin, but also we work a lot with spinal cord injured. We do intrathecal administration in combination with other therapies like cell therapy. That's how we are working on that with a group here in the institute. So for doing all this, we have selected in our hands to work with Polymer Therapeutics, that differentially from other type of nanomedicine like nanoparticles, nanocapsules, or liposomes, we do have a lot of chemistry involving on that, because we use a lot of bio conjugation approaches. So at the end, we are working with polymer conjugates, and we need to treat them as a prodrug for people who are working more on the small molecule area. So it's like a prodrug type of macromolecular prodrug activation. So we work with Polymer Therapeutics. Depending on the cargo, we have a different name, different family. We have polymers that they have active in an activity like Copaxone, for example, that is for multiple sclerosis, or we have polymer protein conjugates like many pegylated proteins, or we have polyplexes, but today I will focus on the polymer drug conjugates as examples for you to share with you, or some examples from the lab. And when we design polymer drug conjugates, especially polymer drug conjugates for application in combination therapy, for small molecules, what is important is to select adequately first the carrier, the polymeric carrier. And this polymeric carrier needs to be multivalent because we need to enhance significantly the loading of the drug. This drug should be linked to the polymer through a bioresponsive linker. And this bioresponsive linker is really a key feature for modifying the release kinetics. And this release kinetics, it can be as a single agent or as a combination, because doing combination therapy is the only way that you secure both drugs in the same cell at the same time in adequate ratio, or adequate release kinetics is really making differentially going better for a person or the other. And also, if you want to go to a specific organelle, to cross a specific biological barrier or a specific receptor, we can add this active targeting moieties, for example, to go to the brain for brain metastasis. If you go IV, we need to put here a peptide that is able to cross the blood-brain barrier, like the AngioPep1, that help us to act as a Trojan horse and to pass by this barrier. So we selected polypeptides, as you expected, because we really like them. We really like to work with polypeptides. They are biodegradable materials. They biodegrade in endogenous metabolites. So this is very, very important when you are thinking to in applications like it has a chronic dosage schedule. For example, neurodegeneration, it's very important to have biodegradable system that it really gets to endogenous metabolites. The chemistry that we have implemented is really well controlled. So we can get very high molecular weights, they are high water solubility, and they have demonstrated in the clinic the safety, and they are more and more going to more hybrid architectures where you can really play with different things. But to synthesize polypeptides, you need to be careful also, because it's a chemistry that has been reporting for many years, in the 50s. But if you really look deeply to what is happening when you use an NCA monomer to implement this ring opening polymerization, you can have many side reactions. So to really have a good polymer chain where you can use as a carrier, you need to trigger a leading polymerization. And for that, we need to initiate the ring opening polymerization and activate this normal amine mechanism to have a control on the starting and the end terminal, and also how many side chains you do have. To do that, we need to have a very clean monomers. We need to have a very controlled ones. And many years ago, when it was the starting point for PTS, based on a SLAB method that it was using chlorhydrates, we use BF4 salts as non-nucleophilic initiators that allow us to trigger this controlled leading polymerization, the NAM mechanism. And also, one of the limitations that was at the time is that even if you control the molecular weight, we are able to reach only a specific DP. So not very large polypeptides were able to be done in a large scale, and also with very nice or narrow polydispersity. So this methodology, this starting synthesis, allow us to go to very high molecular weight. At that time, it was impossible, and also allow us to go to even multi-gram scale that it was not. It was limited at that time also. So we decided to set up PTS. PTS has now a multi-kilo scale plant. They have also implemented GMP facilities. They are working with pharma companies and so on. So they are validating this approach, and many others that they are also improving. But what is really helpful for us, I think it's a really win-win situation where we know that it's important when we transfer something to them based on polypeptides, you need to do it in a proper manner with the controls in place and to the quality of the chemistry and the process that they can really scale up and manufacture to be able to reach a final goal. And also for us, it's really important to have a company that is able to scale up and manufacture something that we believe is interesting therapeutically, because it secures that we could probably at some point reach the patient. So this is a synergistic situation where we are working together in many different aspects. So they managed to get this very well controlled. They are producing as a CDMO, but also we are exploring the possibility of managed polypeptides in different applications. And one of the things we like to work a lot is in different topicity, different architectures. So with linear polymers, we work a lot, but also we work a lot with star-shaped polyglutamates. And what we get when one in front of the other is very different, because the conformation you get is different. And for example, with the star-shaped polyglutamates, you can really fine-tune the size because they self-assemble much easier than the linear ones. So you can get with a star-shaped system, and we like a lot working with polyglutamates because for therapy, small molecules, they are safe and they are anionic, they are taken up by the cells very quickly. So modulating the concentration, we can change the size, and at the same time, you are changing the net zeta potential, because when you think that there's a polyanion, so you are increasing the amount of polymer, so you go to a very high negatively potential. When we got these aggregates, they were disrupting very easily with ionic salts, not with many ionic salts, and we didn't understand really what was going on, because at the beginning our idea with this was we were trying to get longer circulation times in the blood, and we thought that having this BTA we were going to trigger this supramolecular assembly, but it was not correlating with the cryo-TM tomography, because this is not an order in accord. It's a very fluffy type of structure, and also the circular dichroism showed a shift in the minimum that shows that it was not because of the hydrophobic interaction, it was because it was a differential interactions with the branches. So we end up at the end understanding that what was going on, it was a physical behavior that it was described a few years ago by a physicist called Muthu Kumar, that is called the ordinary extraordinary transition behavior, where when you have a high cloud dense of ions, we have a polyanion, and then we have as a contranion we have sodium salts. So this dense cloud of electrons surrounding, they form a very strong transient dipoles, and these transient dipoles nicely stabilizes the structure. So they really is, even if it sounds counterintuitive because it's an anionic system that go together, it really happened because of these dense clouds that they form in this. So this allows us to be or to have with a very versatile system where we can, working with the concentration and also the buffering of the solution, we can move from 4 nanometers to 100 nanometers. If we play with the unimers of 4 nanometers, we can really change the size and the shape of the aggregate, and we can also stabilize them to have something that can go to circulation. So there was really the starting of a new patent that we also licensed to PTS, and they are exploring this type of materials to other applications. And what with Alex thesis, for example, we understood that we could also be very versatile with the size and the shape. So if we change the BTI core and we put something more hydrophobic and play with the length of the side chains, or even with the contranion that we have in the surface, we can end up with things that look like very nice fibers because they are interacting supramolecularly. And they are also with the contranion as an ammonium, for example, you can really push this extra stabilization as a fiber. So we are using this in a spinal cord injured. We can have discrete rods when we have, for example, 25 glutamic acids in a branch instead of 50 that we have this original example I showed you at the beginning. And in this case, we have only 10 glutamic acid. So we are favoring more hydrophobic core and more hydrophilic branches. So you can really change from a fiber to a collapsed nanoparticle. And then we have a very nice chemical tool, understanding this different size and shape, hydrophobic-hydrophilic ratio, the contranions that we can play for topical, for systemic administration, and depending on the application we are looking at, we can also have a go with the different type of self-assembly or supramolecular assembly. But not only playing with this, also with the modification, if you take, for example, the 50 glutamic acid moieties of the original nanoparticle that I was showing you, and you introduce thiol groups. So we can really modulate and we can trigger going from unimolecular micelles of 4 nanometers to nanogel and micron size, because this thiol also triggers different type of self-assemble. So we can really modify in the branches. We can also modify significantly the size of the system, and we are using these nanogels for intranasal delivery also to reach the brain. So this is a beautiful and at the same of like a nice basic chemistry where you can really play with the size and the shape. But if we want to move forward and to have application systemically, we need to stabilize it, and we need to really make that this aggregated self-assembled system are stable under ionic situation where we have, for example, in any physiological fluid. So we design a bottom-up approach where we introduce orthogonal chemistries in the universe. And these orthogonal chemistries allow us to have a cross-linking strategies where when we have already all this strategy here as a self-assembler in the size and the shape that we want to, we cross-link and we stabilize the self-assembled behavior, and then you have a system that can be circulating for a very long time if needed. So this is a highly versatile bottom-up approach where you can put the drugs before or in the surface, depending on how fast, how stable is the drug, how well exposed you want to have them. For example, if you put antigens, it's better if you put it in the surface or some probes, or you have some drugs that you want to release very slowly, it's better to put it before the cross-linking, for example. So this is a very versatile system that once cross-linked, as you see here, which is a model system with click chemistry that we use for vaccines because they maintain for longer the larger size, we have non-modification of the size and non-modification of the zeta potential either, even with the presence of ionic strength. So once stabilized, then the system is stable. But think that we have a system that is degradable. So the PG polyglutamates are degraded by cathepsin B, so it will be degraded up to endogenous glutamic acid. But also, if these cross-linking strategies you put here, that hydrazone, bisulfite bonding, the pH, the redox potential also can destroy, and you can move from 100 to 4 nanometers. So imagine that you have something that is 100 nanometers that gives you a very nice circulation time, and when you reach a tumor, you want to penetrate deeply in the extracellular matrix of the tumor that is acidic. Then you break down these cross-linking bonds, and then you end up with something that is very small that can better penetrate deep in the tumor. So you have a bimodal type of dynamics that you can play with. So we have, this is like a summarizing these possibilities, and we are exploring some of them depending on the design of the system. We derivatize the surface when we want to go for targeting moieties or for nanovaccines, presentation of the antigens. We are putting the drugs in alone or in combination before or after self-assemble, depending on how fast or how quick we want them to be released from the polymer. And why is so important the size and the shape of the system? So for example, if we compare a linear glutamic acid polymer with a star-shaped BTA starting with 50 glutamic acid moieties per branch, compare with the cross-link one, where we have around 750 glutamic acid unimers self-assembled. This is around 4 nanometers, around 8 nanometers, and here you have around 120 nanometers, from 80 to 110, more or less, depending on how we derivatize them. So you end up coming just a half-life, less than an hour circulation to more than 4 days circulation time in the blood. We have been working with Bill Zamboni to understand why these cross-link materials have 4 days circulation time, and they are very stealth to the MPS system. Because think that we are working with polyanions, so they are very hydrophilic. We have done many studies as protein corona. We are not having proteins absorbed in the surface at all with these polyglutamates, unless we are adding drugs and we put hydrophobicity in the surface. Then yes, then the proteins get absorbed. But if the polymer alone, as a naked polymer, they are not. But only when there is like an insult in the circulation, that you have like a toxin in the middle, like Escherichia coli or some acrylates, then the MPS system is uptaking that. So this explains nicely the differential biodistribution. This is a healthy mice, and when we study quantitative biodistribution, all these systems as polyanions, they are excreting renally. They have also suffering, or they are really going through tubular renal circulation, where the larger one is because it's more negative, it gets more. But what it was really nice for us, our finding, is that some of them, especially this cross-linked one, accumulates specifically to the lymph node. So we have a lymphotropism in the system. So this lymph node accumulation has opened up very nice lines of research in the group. We are collaborating with Elena Florindo mainly in melanoma. We have very nice projects with her, and also with Ronin Sachi Fainaro in pancreatic cancer. That they are unpublished data, but we hope to have it there soon, where we have a very differential molecular mechanism triggering not only the production of CD8 positive T cells that are specific for the tumor, but also we are modulating the tumor microenvironment, like diminishing immunosuppressive signaling. So we have a very nice nanovaccines with some antigens presenting there. So I hope next time I will be able to present this project. But just very quickly, one easy manner to take profit of these materials that are self-accumulating or have a tropism to a specific organ like kidneys is to deliver drugs to those organs that you really want to do there. So in collaboration with a group in Fundación Jiménez Díaz, they are a hospital that they are experts on renal diseases. They wanted to have an anti-inflammatory natural drug conjugated to the polymer, what is curcumin. So we conjugated the curcumin to the cross-link system. We did it afterwards because the curcumin is a very unstable drug. So the conditions for the cross-linking in this case were not adequate for curcumin. So we have the cross-link material that it was afterwards derivatized with the curcumin, and we, in a model of acute kidney inflammation triggered by folic acid, what it was really nice to find is that after administration, at the administration, we have very nice reduced levels of urea after the conjugate administration or creatinine. Also, we were able to diminish any signal of apoptosis or reductive or the activated oxygen species, that it was also very nicely reduced when you have the conjugate there. So that was done because curcumin was an anti-inflammatory, antioxidative drug, but our system is targeting this drug into the kidney and acting there as a depot because it gets internalized, it gets renally, tubular renal cells are taken out, they are distributing, degrading slowly, and the drug is released slowly. So you have the drug bioavailable for longer time. So you have an increase in the activity. Similar case with this peptide that this was work when this was my first project that I worked on when I arrived to Valencia, and we got a peptide that these proteins were licensed to a pharma company in Spain, and I was working with Quique Pérez Paya in a group working on protein-protein interactions. So we were working with drugs that they were disrupting the apoptosis formation, and these APAF1 inhibitors, they were if conjugate to the polymer because of this renal tropism show an advantage in front of the small molecule, because this is an experiment done in porks, in swines, where you have a hot ischemia. This is a normal kidney, and you have a kidney after 90 minutes. Ischemia is like when you remove the kidney and you have it in warm, and then you previously administered the conjugate, and you see in the kidney that there was much less signal of cell death, apoptosis, you see here, and it was almost inhibition of caspase-3 activation in the presence of the conjugate of the small molecule because of this beta delivery in a way. So this was licensed, and they are now starting with some derivatives for these APAF1 inhibitors for applications, otica diseases, especially. I think they are starting in that direction. But apart from the polymer and the tropism that the polymer itself is giving us, also we can fine-tune the availability of the drugs that we are conjugating depending on how we do this chemistry, how we do this linking chemistry. Linking chemistry is one of our major expertise in the lab, and also is important when you have more than one drugs in the polymer. So when you are working on diagnosis, you don't need to release the probe. When you are working in therapy, you need to introduce linkers that are break down and releasing the drug to really trigger this therapeutic output. So one project that it was a European project, we are part of it. It was on imaging. We are developing protease-specific probes, and we are conjugated substrate for specific proteases to the polymer. And the idea with the polymer was helping to go to the tropism that was necessary. So we work with different proteases that were overexpressed in tumors and some proteases that were expressed in other diseases. And I will tell you now the story of the osteoarthritis, because with metalloprotein 13 is a metalloprotease that's highly specific when you have this is the osteoarthritic, and there is in collaboration with the Oxford University and also with the CEA, with France, with Vincent Dye, what we did, it was with this probe, it moved a little bit, so the SP14, that it was a peptide substrate of metalloprotease 13, we optimized this linking chemistry to have the adequate spacer for the enzyme to be to have the accessibility in a specific manner to the substrate. So this is the kind of an schematic, one of the derivatives that we did. So we have the polymer, we optimize the spacer, and we have a FRED probe that it was the way that we managed to get this monitor. And this is the peptide substrate for metalloprotein 13 that it was synthesized by Vincent Dye. And we have here the two parts of the FRED probe. So if you see the free substrate combined with the conjugated one, we have even increased in the specificity of the metalloprotease 13 and in vitro test. But what was more important is that we have a very different biodistribution also. So in an in vivo model that it was with Hideaki Nagase in Oxford, it was developed, it was intravenous administration, not intraarticular. So after intravenous administration, with the conjugated probe, we were able to really accumulate in a sustained manner for longer time to have this visualization of the presence of metalloprotease 13. And it was highly specific, and it was at least 2 weeks compared to the free ones. You see here already at 6 weeks, we were able with the conjugate to have a significant imaging compared with the free probe that it was only at 8 weeks. And that was highly specific because we were working also in this project with Sanofi, and Sanofi had a molecule that they were a specific inhibitor of metalloprotease 13. And if we do this imaging analysis in presence of this inhibitor, they quench completely the fluorescence. So it was no cleavage of the substrate, it was not fluorescence output. So we can use it for diagnostic, but we can really also monitor with this diagnostic probe the improve in the health in the animal, for example. So if with the presence of the inhibitor that it was diminishing the presence of metalloprotein 13, we had a diminishment of the signaling, but also an improvement of how the knee it was recovering. So we have also able to monitor the activity of the drug itself. So that was really an application where we have a non-biodegradable linker, enough space and with conformation for accessibility of the metalloprotease 13 that really makes an improvement on the diagnostic part. And now, because I think I'm speaking a lot, but we are linking chemistry for therapy, especially when you have a combination therapy, is very important, and for us is the way to go. As if you don't stop more than one pathway at once, you are not able to really stop the tumor to grow. If we go to metastatic tumors, you need to really do all these complex inhibitions. So that we have already an approved liposome. It's called Combiplex technology in the market, that is now by Jax Pharmaceuticals, where you have a combination of drug to a specific ratio in a liposomes, and it has been declared like much more efficient than the single drugs per separate. So we are having this plus conjugating the drugs through a bioresponsive link into the polymer, so we can fine-tune this release kinetics. I can really fine-tune the release kinetic of one drug in front of the other, and we can really go towards a more personalized approach. But also we are complicated in our life, and we need to implement more physicochemical characterization techniques because the system is more complex. So we apply this in many projects, but in breast cancer, for example, we try to, I'm using this project to show you how we think in the lab. So to design these combination conjugates, we wanted to know which polymer conformation is the best for each subtype. We have to select which drugs are the best for each patient subtype and which biomarkers in each patient subtypes we could use to really better design those linking chemistry. So in this project, what we have done is selecting the combination of drugs. One of the drugs, we are targeting the release of exosomes for tumors. So we are targeting these metastatic processes because we know that the exosomes are one of the responsible mechanisms to transform a metastatic niche for these distant metastases. So if we stop the release of these vesicles from the cell with some mechanism, either by the biogenesis of the release, we could stop the metastasis progression. So we are looking for one drug that was blocking this mechanism of metastasis, and the other drug, one that was already clinical trials or approved in the clinics. So because we are part of the Eric Eupen screen, and we have a screening facility here in my lab, so we were able to screen some libraries of drugs and identify which drug combination were the best for each patient subtype, and also characterizing each patient subtype to look for triggers that allow us to design better the linking chemistry to use in each of the cases. So how we select the drugs is important, the models. So we use 2D models and we do normalized C50 values. So we have systems like this where we look to the Bliss model with the combination index to identify the best combination of drugs that are synergistic in the different subtypes. But because we are focusing on metastasis, we are moving to 3D models. From the most simple one, where we just use the aggregates with the non-adherent plates, and we discriminate those positives of 2D versus the 3D models. So we are, because the 3D models are more enriched on stemness and this metastasis progression, we are the first stage on focusing on those combinations that are focusing on the metastasis regarding the cell death. Also, validating it in a more complex spheroids models where we have not only the 3D but also the interaction of the cells with the matrix. So we have a spheroid model in matrix gel of the different subtypes, and we characterize nicely all of them. And we are using also this strategy to have patient-derived organoids. The difference between the spheroids and the organoids is that the spheroids are built from commercially available cell lines. So we have one cell and all is homogeneous. So we have the spheroid with the same cell. Those derived from patient, it can be representative of heterogeneity because we take the biopsy from the patients, we digest it, and we make them grow with specific growth factors and so on, because the cells are differentiating themselves to form these 3D models. And we have now a biobank of organoids derived from patients that we are using in different projects. When we have selected all these and we have characterized them, I didn't show you the data because it's not published yet, but we have identified which are the condition of each of these 3D models. So for example, the triple negative have a specific redox behavior, ROS behavior, protease behavior, the HER2 positive. So we are taking all these concepts and implementing to those linkers that we need to be using for the selected drugs. And then also when we move to validate it finally in vivo, as you see, we fully characterize our in vivo models. This is two of the ones we have for breast cancer. We have a murine model that is immunocompetent, so for anything that the immune system is involved is better. But this model is very aggressive, and at day 3 we already have metastasis. In the 231 model, the luciferase model is a human. We have metastasis only at day 28, but also we have identified a biomarker of secretion in blood that we can use to monitor the disease. So both of them are very well characterized, and we are using them to validate all the system. Regarding the exosomes, we are also implementing high-throughput screening approaches where we are monitoring by alpha screen technologies the release of exosome, and by immunocytochemistry the biogenesis of the exosome. So we select drugs by IC50, validate that combinations, looking at how these combinations affect this exosome biogenesis or release. So we have graphs like this, where we have expression of exome release by looking at this alpha screen technology, where we are looking at vesicles that have positive at CD9, CD63 that are the markers of exosomes. So we're quantifying how much are released in the cell, and also how much of those are generated intracellularly. So we can select both of them together with the system. Once we selected the drugs, we design the linkers. The normal ones in the mechanism to internalize the cells or the drug, is lysosomotropic delivery. So we are internalizing through endocytosis, going to the endosome and arriving to the lysosome. And in the lysosome, for example, depending on the cell type, but usually we have a very acidic pH, and depending on the different proteases, like cathepsin, for example, is one that is present in most of the tumor cells in a high extension. So thinking that what these differential conditions intracellularly and extracellularly, we can design systems that can be activated only intracellularly or at the tumor microenvironment and not in the blood. So this is an example that we published a couple of years ago, where we combine an aromatase inhibitors, endocrine therapy, together with a chemotherapeutic agent, and we optimize the linking chemistry. We use protease labile linkers, because we wanted to specifically going inside the cell and be degraded in the lysosome, compared with those that can be triggered by pH, like acidic pH. And this acidic pH, it is inside the cell, but also in the tumor microenvironment, you already have an acidic pH. And if you release the drug that is inhibiting the exosome release already in the tumor microenvironment, you are going to be much more effective regarding metastasis. So the idea is that if you want to target metastasis progression, it's better to those drugs that are inhibiting exosome release already at least part of them in the stroma of the tumor. So the first thing we demonstrated, it was that both drugs in the same polymer, this is the aminoglutamide, that is the endocrine agent, and the chemotherapeutic agent doxorubicin, we optimize a linking chemistry for protease labile linkers in the same polymer. They have an effect, a much stronger effect in this 41 model than when we have the two drugs or two conjugates per separate. So having both drugs in the same polymer is important. The ratio of the drug is also very important. He put high loading, low loading. So the high loading of the endocrine agent here was important to introduce a much greater tumor reduction. The tumor accumulation, the plasma half-life, and the differential organ accumulation, this is expected for all the conjugates. So you have a greater tumor accumulation by passive targeting the APR and so on. But also the half-life is greater with the conjugate. And for us, when you are playing with doxorubicin, once you conjugate doxorubicin to a polymer, you avoid hERG accumulation, and then you avoid any cardiotoxicity associated with the anthracyclines, what is very important also for the safety issues. We changing from the protease to the pH labile, and we also wanted to explore how the release kinetics of doxorubicin versus ATM was affecting the activity. So we designed a shorter versus a longer linker. So here is more accessible, and the release of this doxorubicin from the EMCH linker is faster. You have here, we characterize completely the systems very nicely, and what we found is that in each of the linkers, the shorter or the longer linker, it was more effective than the protease labile. Do we have safety? No issues. We have the survival was perfect. No cardiotoxicity at all. So this is a free doxorubicin. It was cardiotoxic. The conjugates were not cardiotoxic at all. But when we look at the hepatic inflammation, we found out a slight hepatic toxicity with a longer linker, because we have a very fast release of doxorubicin. So we have this small inflammation, hydrophic degeneration in the liver. So we wanted to avoid this possible safety issue, and we moved to the shorter hydrazone linker. So with the shorter one, we did the metastasis progression. So this is the model for metastasis progression. And we take out the lungs metastasis. This is the histology of the lungs. And you see that with the conjugate, with the shorter hydrazone linker, we managed to really diminish up to 94% lung metastasis. This was not happening with the protease Laval, because we need the tox to be released in the already in the tumor microenvironment. And when we analyze why this is happening, is really all the mechanisms that the exosome release is acting at. So like cell migration, proliferation, metastasis, angiogenesis are down-regulating, and we are up-regulating processes of apoptosis, autophagy, and we have really an activation of the immune system. So we are really acting at the tumor microenvironment level. And if I have 1 minute or 2 minutes left, just to show you, if you need to cross a very challenging biological barrier, we need to use this active targeting for the CNS, for example, delivery. And we have projects on the CNS, different applications, also IV and intranasal, but talking to brain metastasis is important to all this breast, and then you add a peptide that is triggering this brain metastasis progression. So to go to the CNS, if you go IV, you cross the ABB using this Trojan horse mechanism. That is the one that is working the best. But also we are exploring the nose to brain, and I hope to show you this in another occasion. And we are working on AngioPep2, another peptide. That's why I wanted to show you this data, because AngioPep2 has a 19 amino acids. It's already clinics conjugated to paclitaxel. Is in phase 2 clinical trials for glioblastoma. And the LRP is targeting the LRP1 receptor that is overexpressed when you have brain metastasis of breast cancer. So it's overexpressed here. So Fernanda's thesis, and we are collaborating with Manuel Valiente in CENIO, he synthesizes a combination of drugs with this AngioPep, and they managed to really bypass the BBB to reach the brain. And this is a model that Manuel Valiente has, where you have GFP labeled 231 cells, breast cancer cells in the brain of the animal that are in green. And you see this is the blood-brain barrier, the blood vessel. Our polymer is Cy5 label. And you know how they are able to bypass and colocalize with the cells. And why how it's working, they are not only sticking in this is the histology of the brain after IV administration, is not only sticking in the vasculature and distributed to the neurogenesis areas, and also is going distally from the blood vessels and are internalized and colocalizing with the lysosome in the cells in the brain. So we are having a different distribution in the brain, capable to cross and also to act as a depot. And this helps us to validate. The first validation, the publication we have with this is in an Alzheimer model. So we conjugated non-toxic drugs, we conjugated anti-inflammatory drugs, and antioxidant drugs like curcumin. We did also screening and so on to identify those drugs. So we selected two of them, like curcumin and this MAO therapies, and we demonstrated that we are able to diminish in organotypic cultures the inflammation triggered by the A-beta amyloid in the presence of our conjugate. Also that our conjugates, our polymer per se is active because it's interacting with the fibers, but also you see here with the NMR that we are able to disrupt the fibers only with the polymer, but also we are able to have a therapeutic window that is very safe even after repeating administration, because we are in a chronic administration in the animal, so we have no issue with toxicity. And we have very nice data in an Alzheimer's model where after this repeated administration, we managed to enhance significantly when we conjugated a combination of drugs, the biscurcumin or genistein. We have validation is several combination of drugs compared with the free drugs in the recovery of the olfactory memory. when a patient of Alzheimer is losing the first is the olfactory memory. So because of this differential distribution in the brain, especially accumulating in the olfactory bulb and the hippocampus, that is the neurogenesis sites where you have more vasculature in the brain, it really helps to release the drug there and to have a better result on recovery for these neurodegeneration approaches. So with this, very quickly, I hope to have shown you many information and examples where polypeptides, and I'm showing you data with polyglutamates, but we are also working with polyornithines, with polyprolines, with polyhistidines. We are working with many other polypeptides, and we need to rationally design them, and then you can reach really nice therapeutic applications. So with that, showing you the people who is really working in the lab. So they are really nice, a bunch of people working a lot, very interactive, from chemistry to biology. I have many collaborators for many of the projects and funding agency, and thank you so much for your attention. I'm happy to take any questions.
Yes. Thank you very much, Maria, for sharing your results with us, at least some of the many results you had in the last years. During your talk, we collected some questions, and the first one came up quite early, and I guess you already answered quite a bit of it. Which polymers do you recommend for passing the blood-brain barrier? I think one cannot generally say this, but what's your recommendation?
For the crossing the blood-brain barrier, depending on the administration route that you are selecting. So if you select the intravenous administration route, you need to the polymers that there are some people working with polymers, and there are some biotech companies in Spain, for example, that they are using polymers based on polypeptides, that they are passing the BBB by passive mechanism. But the most used approach is using targeting moieties, as the one I show you, that they are using this receptor-mediated transcytosis. This and one that is working the best is the LRP1. The polymer to be associated to that is the most important is the targeting moiety. And then, depending on what you want to deliver, you design your polymer. So if it's a small molecule, polyglutamates work really nicely, but also there are other type of polymers that you can also work. Mm-hmm. You are going to the intranasal route. The targeting moiety is not important. It's more important to have a polymer that has a mucodiffusive, mucoadhesive properties. You need to go nose to brain. And also the polymer needs to have these charges. That's why we like a lot of polypeptides, because they need to diffuse through the brain and they need to travel through the neurons. So first you need to have mucoadhesive, mucodiffusive properties, nose to brain. And when you arrive to the mitral neurons, you need to have this differential potential properties to be able to migrate to the brain and diffuse homogeneously. So they are different, depending on the delivery cargo you want to, and depending on the administration route, the design is very different.
Okay, thank you. Then one rather technical question Thomas asked, is it possible to synthesize amino acids of molecular weights of 100 kilodalton or even more? Is it technically possible? And on the other hand, does it make sense at all, or do you say it is possible, but for which purpose?
The 100 kilodaltons, we have synthesized 100 kilodaltons and even more, because when we wanted to demonstrate, that is published in the Polymer Chemistry paper 2013, when we validated these BF4 salts as initiators. One of the things that we wanted to demonstrate is that we wanted to go to very large molecular weight, that it was not possible before. The larger the polymer, the more time is taken. If it's important to have a very big molecular weight polymers, at the beginning we thought it was. In fact, PTS is doing 1 million daltons as a start brush system. So we are using them in nanovaccine delivery, for example. So it's possible to reach very high molecular weight, depending also on the architecture. Is worth this molecular weight high, depending on what you want to. And also you can reach the same molecular weight or the same sizes using this self-assemble or these supramolecular assembles or a kind of aggregation behavior. If you have a greater molecular weight, you have longer circulation times. But sometimes when you have a very big molecular weight, depending on how compact is your molecule, maybe it's not good for your purpose because the degradability goes down and it's not that much biodegradable. If it is too globular, maybe it's excreted faster and it's not going the way you want to. So it needs to be, depending on the application, it needs to be. It's good to have larger sizes for intranasal. So you need to have larger than 100 to go intranasal. That is working better. This is not different. But systemically, the size, it cannot be more than 100, 110 to really go to where you want to go, not go into the liver or not go into the spleen or the kidneys very quickly.
Okay, thank you. The next question that came up when you were showing the half-life extension effect with the star-shaped polymer compared to the linear, did you also check the half-life extension effect when cross-linking linear PGA?
No, we didn't. We could do, but the problem is we need to, we have done, for the intranasal delivery, we have linear cross-link with thiol groups, but it was not the same click. No, it was not exactly the same structures that with the star shape. So we haven't done it. So I cannot answer for how long. You can still have it, but you have to force a lot the concentration. So, for example, to do the self-assembly with the star system, usually at 2 milligrams milliliter, you already have a nice concentration for the self-assembly. With the linear polymer, you have to push much more. You have to go much higher concentration to really trigger this self-assemble behavior. So it depends on the topicity. The concentration needed to have it closer chains to cross-link is different also. But no, we haven't done it, so I cannot answer. We didn't compare directly. But possible is possible. It's possible.
Okay. And then one question coming in when we were showing those breast cancer organoids from patient-derived cultures. So how long does it take from tumor biopsy to the development of such a organoid screening model?
I have a PhD student devoted to implement all this model in the lab. So it's not trivial. We are using, we have a collaboration with the clinicians. We are taking the biopsies. We are processing the same day, and to make them grow and to establish, it depends on the amount of samples you are getting and the aggressiveness of the tumor. All of them form spheroid organoids at the beginning. All of them start growing. But what is the limiting step and where the success rate is coming to is that they are really able to be frozen down and to proliferate afterwards. So you need for them to grow, it takes weeks, like up to 3 weeks, I think. We have this biobank establishment that we have 20 that we managed to amplify and froze down, and then we defrost to do the experiments. It really took months to have a proper established organoid. The success rate for us, it was around 40% to establish the organoids and to have those enough amount to do the proper testing of therapeutics with all the amount of groups that we do have, like having millions of those. We only have around 20% of the samples that we managed to get a good storage to have this testing. So implementing this biobank, now that we have the protocols established in breast cancer, it's taking weeks only, like in 1 month we could have it. When you start with a new polymer or new tumor, it depends. So we are already starting also with prostate. They are much more complicated than breast. We are also starting with ovarian. Ovarian are more or less similar to breast and even better because you can have more tissue for the patients because it's a larger area, so it's less problematic to do that. But also it depends on the patient subtype because the aggressiveness of triple negative, for example, is much more than luminal A. So triple negative breast cancer, the success rate of implementation was much greater It was like 3 times better than the luminal A, that is grow much slower. But it reflects also patient heterogeneity in that sense.
Okay, so one question that is comes after this to the star-shaped PGA that allows a better half-life. Is it a general rule that this allows a better half-life, or only valid for the PGA? Does it work similarly with star-shaped polylysines, for example?
Polylysines are positively charged materials, for example, and we have an example on dendrimer lysines. Polylysin as a dendrimeric carrier that is in phase 2 clinical trials with Star Pharma from Australia, in collaboration also with AstraZeneca. But they cannot go naked. They cannot go as a polylysin because as a polycationic, they are hemolytic. So what they have done is they are pegylating the surface, and they are also conjugating some drugs on the surface. So the zeta potential of the surface is almost neutral to avoid this hemolysis. So what is it can be generalized as a rule is like independent of the material, this is neutral or negatively charged, is safer for systemic administration. Positive charged material are more toxic because they are interacting stronger with the membranes, and they can be hemolytic and breaking down this, and that's really triggering either a huge immune response or hemolysis problems. And also, if you think on neutral or negative charged polymers, larger molecular weight, larger sizes triggers longer circulation times because the fenestration for the excretion are certain size, so you have more time circulating because they are larger, so they cannot fenestrate that easier. So you have something very small, 5 nanometers, it will be gone very quickly in minutes, or in but if you have something that is around 100 nanometers, 80 to 100 nanometers, it can go for a much longer time. And if it is stealth and nobody recognizes, it stays even longer.
Thank you. Then one thing, are your particles being uptaken by macrophages or dendritic cells? And are there any attempts on the vaccine field?
Yes. In fact, the project I didn't tell you about with Elena, what we know is that this cross-link polyglutamate system is uptaken by dendritic cells in a very selective manner. And this is the reason why we managed to get this 40% uninjected dose in the lymph nodes. So we have a lymphotropism because we have an active targeting to drive the polymer to the lymph nodes. And there, they trigger maturation in a cross-presentation manner. So we have overexpression of CD8 and CD86 that triggers the release of the CD8 positive T cells that kills the tumor. And also we have other mechanisms there. But yes, our polyglutamate cross-link are taken by dendritic cells, but also it was demonstrated in the clinics already that Opaxio, that is a PGA plaquitac cell, for example, that it was in phase 3 clinical trials, the major contribution for tumor activity was coming by the tumor-associated macrophages, because the conjugate were uptaking much faster by tumor macrophages that were digesting the system and then releasing the free drug. And this is something that very nicely the group of Miles Miller in Harvard University or the hospital is really showing that if you have infiltrates of MPS system in your tumor, they are really contributing in a very nice manner for the activity because if they are uptaking, they are increasing the percentage of targeting the tumor because the capability of uptaking for these macrophages, they are by phagocytosis or macropinocytosis is much faster. They accumulate much more nanomedicine than those tumors that doesn't have any infiltrate in there. And there are nice papers from this Miller group that shows that these infiltrates really helps on tumor anti-tumor properties of the polymer. So yes, targeting the macrophage is a good strategy to enhance this possibility.
Okay, and one last question I currently see. Is it possible to predict the effects of linker chemistry and linker length and cleaving mechanism on in vivo behavior? Because you showed that protease versus pH label linkers and spacer length and so on, or is it rather a thing of trial and error? So can you predict or
we are one
interpret the results afterwards?
One of the things that we are trying is this is the biomarker, functional biomarker process I was mentioning. We are trying to implement techniques like quality by design, design of experiments, to really narrow down the possibilities. It cannot be trial and error because if not, we will never finish. But what we have done is we try to understand which are the overexpressed triggers in each of the cells, so models that we need to try. So if we need to go for a redox, if we need to go for a pH trigger or a specific protease trigger, so we select those. And we know by heart, because we know the system, that is the accessibility is really making the release differential. And we need a certain percentage of the release of the drug to have activity without toxicity in the models that we do have. That we know because we have done a lot. But we put all the data in this design of experiment, and we have kind of a narrowed down possibility. But this is where these people working on artificial intelligence, data mining, and things like that are really helpful, because I would love to have a predictor that tell me, for this model, you have this combination with this linker, with this release kinetics, because we try to do that. We have some predictors, but not full of them. So we still have a family of 3 or 4, and then we select the best one. We don't know if this will be the best selected, but at least we narrow down from the very beginning to now. We are much directed to the point. So it's not a trial and error, but still we are not there with a proper graph where it says in this area is the area you need to design. But I'm working towards this. I'm very far because I'm not an expert, so we're trying to get something onto that area also to have a better prediction.
Okay, thank you, Maria. Thanks a lot for sharing your results with us. Currently, I do not see any further questions. Obviously, people really liked your presentation because we did not lose participants until the very end of the session. This is a very good sign. So Maria, thanks again. It was a pleasure to have you here.
Thank you very much for the invitation. My pleasure. Okay, see you very soon. Bye-bye.
Thank you. So with this, we would like to close our online workshop. As a follow-up, we will provide the recordings. Thanks again for joining us. Thanks to both of our speakers, and also a big thank you to our QA manager, Klaus Helm, for taking care of the technical support. We hope you could collect some helpful ideas and information from this workshop, and we are looking forward to meeting you in the next one, probably in September. We will announce it on time on our homepage and via social media. Enjoy a nice summer, stay healthy, and hopefully see you soon. Goodbye.